Today I will discuss some of our work that's related to brain functions on emotional affective aspects of pain, and I will focus on the amygdala, a brain region that has been implicated in emotional behaviors and affective disorders. And specifically, I want to focus on neuroplasticity and the role of neuroplasticity in the amygdala in pain. Neuroplasticity can be defined as structural or functional changes that outlast or become independent of the event that triggers them. And you have heard that is important for normal physiological functions such as learning and memory. But in pain conditions, neuroplasticity in the amygdala is linked to emotional responses and also to the modulation of sensory aspects of pain and to anxiodepressive disorders. What I want to do today is point out that neuroplasticity changes in the transition from acute to chronic pain. And this is very much in line to what Dr. Abkarian told you yesterday, that chronic pain is not simply the persistence, the prolonged persistence of acute pain, but it is something qualitatively different. And we believe also that neuroimmune signaling plays an important role in this transition. So pain is a complex disorder with different components, but it's the unpleasantness of pain that defines it. And so the brain regions that are concerned with emotional behaviors and aspects, they receive nociceptive information from peripheral tissues through a dedicated pain pathway that starts in the spinal cord and goes through the brainstem and then feeds into these regions. And so this is distinct from the classical pain pathway, the spinothalamic tract, that feeds more into the thalamocortical systems that are more concerned with the sensory discriminative aspects of pain. What's important is that these brain regions, amygdala and others, also connect to descending pain modulatory systems, and that is a way how emotional centers gain access to the modulation of sensory aspects of pain. So amygdala and other regions, such as hippocampus and hypothalamus, are part of what is called the limbic system that is preserved in the human brain and exists in the rodent brain, and it's important for survival functions such as the appropriate responses to threat conditions. And based on our work and that of others, we have developed this kind of circuitry that involves pain processing in the amygdala. And I think this will help you understand some of the data that I will show you. So the amygdala consists of different nuclei, the central nucleus and the basolateral amygdala. And it is the central nucleus that receives nociceptive information from this pain pathway that I just mentioned. The amygdala also receives more polymodal, including pain-related information from thalamic cortical systems, cortex in particular, the insular cortex and anterior cingulate cortex. And those two lines of information then converge onto the central nucleus. The central nucleus includes a very complicated network of different cell types that some of them I will talk about later. And these form projections to other brain regions to modulate behavioral responses. And I should mention that this pain pathway is unique because it contains a neuropeptide, CGRP. You may have heard of CGRP in the context of migraine treatment. And this processing here is, of course, under a cortical control, such as from the medial prefrontal cortex. One of the output regions is the hypothalamus, and we have more recently become involved in the analysis of that. The hypothalamus can control the release of hormones such as prolactin, and prolactin has more recently been implicated actually in female-specific pain conditions. But I won't have time to do that today. I will focus on the amygdala. So the amygdala includes different cell types. The central nucleus is what we are interested in as the output nucleus. Some of those receive input from this pain pathway that I mentioned, spinal cord, parabrachial area to the central nucleus. The neurons that receive input from this nociceptive pathway are corticotropin releasing factor hormones. CRF is a stress hormone, but also a neuromodulator. PKZ delta expressing neurons also receive input from this pathway. Another type, though, the somatostatin expressing neurons do not receive that input. So it's important to understand the different cell types in the central nucleus of the amygdala. With regard to the output, the projections of these neurons, CRF neurons have long been known to project to various brain regions that are important for behavioral modulation, including the forebrain regions, hypothalamus, and various brainstem centers that are important for pain modulation. Somatostatin neurons also project to several of those targets in the brainstem and the forebrain. PKC delta neurons have more recently recognized as projection neurons. They tend to project more to the forebrain and diencephalic areas. So the system in the central nucleus receives nociceptive information and is also positioned to provide output and modulate other brain regions. So what I want to do today is show you changes, neuroplastic changes in these different regions and cell types. Then evidence that neuroimmune signaling is involved in the transition from acute to chronic pain, and in some of the behavioral consequences. So about 20 years ago, we published for the first time an article on changes in the amygdala. And what we did first is record the activity of individual neurons in the central nucleus of the amygdala. These are electrical signals, action potentials, or spikes. We did this in the intact anesthetized animal, and then we induced a pain model, an acute arthritis induced by kaolin and carrageenan injections into one knee joint. The advantage of that model, it's localized. It develops within a few hours, and so that allowed us to record the same neuron before and after induction of that pain model, and you see that the activity in these cells increased. So to determine whether these changes just reflect enhanced incoming signals from the pain pathways, or if there's an intrinsic component to the amygdala, we introduced the brain slice preparation to the pain field. And in the brain slice preparation, you can identify different inputs and synapses and record individual cell types. And so we took brain slices that contain the amygdala from normal animals and from animals in which this pain model had been induced in vivo. And then we looked at the excitatory synaptic transmission, a measure of excitatory drive onto those cells, and also excitability, the number of action potentials induced by depolarizing stimulus, a measure of hyperexcitability. And what we found is that in brain slices from animals with a pain condition, there was enhanced excitatory transmission and enhanced excitability. So this is a reduced brain slice preparation that is disconnected from the site of injury, the knee joint, the peripheral nervous system, the spinal cord, and the brainstem. So changes must have developed in vivo that are now preserved in this reduced brain slice preparation that justify the definition of neuroplasticity. So this was confirmed by other groups, but most of the studies were really done in acute pain models, various pain models, formalin, capsaicin, arthritis, visceral pain, and in some early stages of a neuropathic pain model up to 2 weeks. So we were wondering what happens at the chronic stage. And the other big question was, what happens to different cell types? Are there differences? I showed you that CRF neurons, somatostatin neurons, they have differences with regard to the input and potential projection sites. So we used a neuropathic pain model, and again, we recorded excitatory synaptic transmission and excitability, first in brain slices taken from acute animals. And we looked at CRF neurons and non-CRF neurons that also project to different brain regions. And what we found is that there's enhanced synaptic transmission, synaptic plasticity, like I showed you in the other slide. So that confirmed the findings, and there was some evidence also for increased hyperexcitability in CRF neurons, but not in non-CRF neurons. And then we looked at the chronic phase and took brain slices from animals that had this neuropathic pain model for 4 weeks. And there was a big surprise. What you see here, there's no change anymore in enhanced synaptic transmission, no evidence for synaptic plasticity in CRF neurons and in non-CRF neurons. So is the amygdala silent? No. The non-CRF neurons still show hyperexcitability, enhanced action potential firing, whereas the CRF neurons don't. So this is a dramatic change from the acute to the chronic stage. The acute stage is synaptically driven hyperexcitability in CRF neurons. In the chronic stage, loss of synaptic plasticity, synaptic plasticity independent persistence of hyperexcitability. And so how can we explain that? We also found that there's not just functional changes, but actually structural changes. So this pain pathway that I mentioned earlier, that forms actually these unique basket-like contacts around these amygdala neurons, and you see them here in the immunohistochemical staining. And these are anchored to the CRF neurons through what is called a transsynaptic signaling complex that consists of 3 elements. We have presynaptically the norexin and postsynaptically a delta glutamate receptor, and they are anchored together with cerebellin. So then we looked at the chronic pain model, and you see that there's these basket-like structures actually have largely disappeared. And that is because the cerebellin is downregulated, the GLUT1 receptor is downregulated, and as a consequence, the postsynaptic AMPA receptors and excitatory glutamate receptor are upregulated. And we can normalize that if we inject cerebellin into the brain. Cerebellin expression is restored, the GLUT1 expression is restored, and the AMPA receptor expression is controlled. So this is evidence for functional and structural changes in the chronic pain model. So to explain that change from acute to chronic pain, from synaptic plasticity dependent to synaptic plasticity independent hyperexcitability, and the shift from CRF neurons to non-CRF neurons, we thought maybe we need to look at non-neuronal elements. And those include astrocytes and microglia that play an important role in synaptic plasticity, in intact synaptic functions. And so we developed this hypothesis that maybe at the acute stage of a pain condition, there's an activation of CRF neurons that couples to neuroimmune signaling, activation of non-neuronal cells, and that maintains then the persistence of hyperexcitability in non-CRF neurons, and this neuroplasticity then maintains chronic pain behaviors. So to study something as complex as neuroimmune signaling, we used an approach that's called transcriptomics, and that is a tool to measure gene expression changes. So we take amygdala brain tissue, isolate the RNA, send it off for bulk sequencing to our colleagues at the University of Texas in Austin. And after quality control, we do a number of bioinformatic strategies to determine differentially expressed genes. And then we do also a pathway analysis and a network analysis. And with transcriptomics, you can compare different conditions, sham versus pain condition, left versus right, different brain regions. And so I want to show you just a few examples. What we did is when we found 570, I think, differentially expressed genes, then you can look up what is the canonical function of these genes, and you can come up with likely pathways that are affected. And you see some that are downregulated and some that are upregulated. And interestingly, we found that a number of these molecular gene markers are associated with synaptic and neurotransmission, and they were downregulated, which is consistent with the loss of synaptic plasticity that I showed you earlier. Whereas other genes that are linked to neuroglia and glial activation and function were upregulated. Here are just a number of examples of these genes. You see the cerebellin was downregulated, I showed you that. BDNF, which is also an important molecule for synaptic plasticity, was downregulated. And maybe this diagram shows that a bit more clearly. This is a network analysis with a focus on this neuroimmune signaling, because we have evidence that they were upregulated. And so you can look at the different genes that were either increased or decreased, and what is their function, inhibitory or excitatory function on synaptic transmission or neuroinflammation. And so basically that pattern suggests that there's evidence for decreased synaptic transmission and increased for neuroimmune signaling. So then we wanted to see what is really going on in the brain. So we did some staining for microglia, this classical staining. We did this in normal brain and then in this chronic pain model. And you see there's not just an overall global increase in microglia activation, but it's region specific. Find it in the amygdala, the insula, all regions that receive nociceptive input. Somatosensory cortex that receives input from the hind limb, which is where this model was induced, but not the somatosensory cortical areas that receives input from the whiskers. So there's some specificity, and that is in support of a pathway neuronal activity-driven activation of microglia. And when we look at the cell more in fine-grained analysis, then you can see that the shape of these microglia at the acute stage compared to naive and sham changes. You see there's an increased number and maybe some hyperramification. And then most dramatically at the chronic stage, you see here in increased signal intensity, the shape resembles that of reactive microglia. And we did a quantification of that, and we found that the density, the number of microglia per tissue, is increased at the acute and at the chronic stage in the central nucleus, whereas the intensity, the signal that may reflect, the increased shape of these microglia is only increased at the chronic stage. So there are neuroplastic changes that are different at the chronic stage compared to the acute stage. Then we also looked at the astrocytes and did some staining here, and you see that there's an increased expression of astrocytes at the chronic stage. With PCR, we found that also. And again, comparing chronic to acute stage, there's an increase of astrocytes. And that's actually consistent with what has been observed in the spinal cord, that maybe the astrocytes come in to play at a later stage. So, and finally, what are the behavioral consequences? So we have a number of strategies to do that, but we do actually run a number of assays in our lab. And for those that are not familiar maybe with preclinical studies, to measure sensory aspects of pain, we use the classical reflex measurements. So you measure the force, the threshold that is required for a nociceptive reflex. For emotional responses, we measure vocalizations evoked by noxious stimuli. For anxiety-like behavior, we measure use the elevated plus maze, open field, light dark box. These are all assays that measure exploratory behavior, and anxiety-like behavior is measured as the avoidance of open arms or open spaces or bright lit areas. We have also more recently added the grimace test, where we measure facial expressions of these animals. These are 5 points that are scored: the ears and eyes and nose bulging and whiskers. And to link neuronal activity to these behavioral outcome measures, we use a number of strategies, but these optogenetics and chemogenetics, they allow a cell type specific manipulation, which is important because I showed you there's differences in the changes of these cell types. But both techniques rely on the expression of artificial molecules that are not normally present in the body, using viral or transgenic strategies. In case of optogenetics, these light-sensitive molecules can activate or inhibit neuronal activity. For the chemogenetics, you express G-coupled receptors, either GQ to activate neurons or GI to inhibit neurons, and then you apply a designer drug. Again, that's a compound that is normally inactive and not present in the body. So I just wanted to show you that the strategy was to focus on these CRF neurons, because that's the hypothesis that CRF neurons are the ones that are activated first and then drive this neuroimmune signaling. So what we did is we used chemogenetics to activate the CRF neurons and then see if that has behavioral consequences and if it involves microglia activation. Okay, so we activate CRF neurons in the central nucleus of the amygdala chemogenetically, and you see that the sensitivity increases, the threshold drops, vocalizations increase, and in the grimace test, the score increases, suggesting that there's ongoing pain. So under normal conditions, normal animal activation of CRF neurons produces pain-like behaviors. To implicate the microglia in that, we used a strategy to deplete microglia, and you see the microglia are depleted from the central nucleus with this application of PLX. And in these animals where the microglia were depleted, CRF activation has no longer an effect. No effect on sensitivity, no effect on vocalizations, no effect on ongoing pain. So that suggests CRF neuron activation can trigger behavioral responses, but it requires the presence or the availability of microglia. So then we looked at microglia, and we actually wanted to see what is the link. How do CRF neurons trigger neuroimmune signaling, microglia activation? So we again went back to our transcriptomic analysis, and one of the molecules that stood out as a differentially expressed gene is this HMGB1. HMGB1 is present in different cell types, and it acts on different receptors, TLR2, 4, and RAGE. These are expressed on, among other things, microglia cells. So we looked at the expression of HMGB1, and we found that it's actually increased in this neuropathic pain model in the right but not the left amygdala. At the qPCR validation, we also confirmed that it's in the right increased but not in the left, and it's in male and females increased. And we confirmed that HMGB1 is actually expressed in neurons. So this is the staining for HMGB1 in the central nucleus. This is co-labeling for neurons with NU-N. And you see all those cells that also express NU-N express HMGB1. And there's a few cells that are not neurons that express HMGB1. So we quantified that. So there's a larger number of cells expressing HMGB1 than non-HMGB1 expressing cells. The majority is neurons, and in fact, there's not one neuron that does not express HMGB1. So that is consistent with neurons having HMGB1, releasing it, and that could trigger microglia activation. And importantly, HMGB1 in neurons is also expressed in these CRF neurons. Here's the co-labeling, and CRF neurons express HMGB1. So then, to link HMGB1 to behavior, we used a knockdown strategy using siRNA, injecting it in the central nucleus of the amygdala. And when we knock down HMGB1, we can decrease the sensitivity, increase the thresholds in different assays in males and females, and we can decrease the vocalizations in this neuropathic pain model. So together that suggests CRF neurons can trigger behavior. They require microglia activation, and through a molecule that is, for example, HMGB1, and if we knock down this link between CRF neurons and microglia, then we can inhibit pain behaviors. So in summary, I tried to show you some evidence that neuroplasticity in the amygdala changes in the transition from acute to chronic pain. Starts with the activation of CRF neurons. HMGB1 is a potential target that could lead to the activation of microglia. And also importantly, the amygdala plays an important role in this emotional behavior, but it can also modulate sensory behavior. So it's really at the intersection of nociceptive inputs and emotional circuits. And so with that, I want to acknowledge people in my lab, and maybe we have time for 1 or 2 questions.